Embryogeny in dicot plants. Embryogeny here means development of embryo. Dicot plants are characterized by presence of seeds with two cotyledons. That's why dicot plants. Di refers to two, cot here means cotyledons. Say for example, if we cut open the germinating seed of gram seeds, then we are able to observe two cotyledons. Generally, cotyledons are footstone. These are foot reservoir which nourishes the developing embryo. Developing embryo in the seeds are characterized by presence of plumule, which will be developing into aerial parts, or these plumules will be forming future shoot system, and your radicle of the embryo will be developing into future root system. So in order to remember radicle forming root system, both the term radicle and root system are starting with letter R. So radicle will be forming root system and plumule will be developing into shoot system. Plumule and radicle are part of embryo, and embryo get nourishment or get nutrients from cotyledons. In case of dicot plants, we have seeds with two cotyledons. That's why they are called as dicot plants. Now, embryogeny in dicot plants can be described as development of embryo of dicot plants. Now, first look at some facts about embryo development in general. We know that embryo usually develops at the region called micropylar pole, where there is development of zygote. So zygote is formed at the micropylar end or micropylar pole or micropylar region. Now, in order to develop zygote into embryo through process of mitosis, zygote requires nutrient, and the nutrients are provided by formed endosperm. Now in plants, in order to form proembryo from zygote, we should have certain amount of endosperm. Endosperm will be nourishing the zygote, and in a way it will undergo mitosis and cell differentiation to form proembryo, which will be developing into embryo, and later on there is formation of seed. So it is adaptation in plants that first there is formation of endosperm, and after the development of embryosperm, then there is development of embryo. So after the formation of zygote, zygote will get nutrient from endosperm, and zygote will undergo mitosis, differentiation of cell, and form embryo. Now embryo is suspended within the ovule of your ovary. Development of embryo in dicot plant and monocot plants are similar in the earlier stages, but these are different if we observe in detail about the later stages of embryo development. Say in case of dicot plants, there is formation of embryo through different stages, and the different stages are characterized by formation of proembryo, which will be later forming globular shaped embryo, and globular shaped embryo will be later developing, undergoing mitosis and cell differentiation to form heart shaped embryo, which later develops into torpedo shaped embryo, and which will be later forming walking stick stage of embryo, and finally there is development of mature embryo. This is very different from monocot. In case of monocot, though earlier stages of development of embryo in monocot plants are similar to that of your dicot plants till the globular stage, but later stages are different. Say after the globular stage of embryo development in case of monocot, we have scutellar development, we have scutellar stage, and after the scutellar stage we have Colliop-Taylor stage. So Colliop-Taylor stage and scutellar development is not found in Dicot embryo. Now let's discuss in detail about the development of embryo in dicot plants with some graphics. We know that zygote is formed at the micropylar end after double fertilization. Now, zygote will undergo mitosis to form two-celled proembryo. The two-celled proembryo is having a smaller cell at the chalazal pore, which is far away from the micropylar region, and that cell is called as terminal cell, and near to the micropylar pole we have a bigger cell formed, which is known as basal cell. Now later your terminal cell will be forming embryo and basal cell will be developing into suspensor cell. So in the quadrant stage of embryo, you are able to find quadrant embryo, wherein we have quadrant here refers to four. So four cells are present, two cells which are seen front and two cells are shown behind. So we have four cells in the quadrant stage of embryo, and your basal cells will be forming suspensor cells. Now these cells will undergo continuous mitosis and cell differentiation to form octant stage of embryo. Octant here refers to 8. Now in the octant stage of embryo, your embryo is having 8 cells. 4 cells at the front and your other 4 cells are shown behind. And we have suspensor cell, which will be helping in suspension of your embryo, or suspensor cells will be pushing your embryo away from the micropylar pole and into the endosperm, so that embryo receives proper nutrition, proper nutrients from the endosperm. And we have a bigger cell at the micropylar pole that is called as hostorial cell, which will be helping in nourishing your suspensor cells. Suspensor cells require nutrient which are provided or which are absorbed by hostorial cell. Later on, after mitosis and cell differentiation, we have formation of globular shaped stage of embryo, wherein we are able to observe globe shaped embryo, which is having many cells, and those different cells are differentiated to form outer region, and those cells which are forming the outer region forms the layer of dermatogen, which will be forming protoderm, and inside the dermatogen we have ground meristem and we have procambium, which is within the ground meristem. So these different cells will be developing into entire new plant. We do have suspensor cell, which usually hang or pushes the globular embryo into the endosperm, so that developing embryo receives proper nutrition. We are having hostorial cell, which will be feeding the suspensor cell by absorption of nutrients from the nearby cells. Later, after mitosis, cell differentiation, there is accumulation of more cells in the embryo, and it enters into heart-shaped embryo, wherein we are able to observe heart-shaped embryo. Embryo is heart-shaped in the heart-shaped stage of embryo, wherein we are able to also find suspensor cell, which will be helping in pushing the heart-shaped embryo in the embryo endosperm, so that an embryo receives proper nutrients. We also have after that, after the heart-shaped embryo development, there is again undergoing of mitosis, cell differentiation, and there is formation of torpedo-shaped embryo. Torpedo-shaped embryo is having shape similar to that of submarine. So your torpedo-shaped embryo is having shape similar to your submarine, right? And we have at the stage of torpedo-shaped embryo, we are able to observe the different regions of the cell, say cotyledons, we have plumule, we have radicals, we are also able to, we do have suspensor cell, and with the torpedo-shaped embryo, we can observe the region of cotyledons. We have two cotyledons in the dicot embryo, and we have plumule, which will be developing into shoot system, and we have uh, radical, which will be forming the root system. Right now, we are able to observe the embryonal axis, the center of the embryo or the center of the two cotyledons, wherein we are also able to observe the uh, epicotyle, hypocotyle of the mature embryo. So this is a walking stick stage of your dicot embryo, wherein you are able to find your embryonal axis, which is having lower region called hypocotyle, and it is also having upper region which is called as epicotyle. Over here it's not shown. Now in the typical dicot embryo, we are able to observe two cotyledons. So we have two cotyledons, and the upper part of the embryonal axis will be having plumule. We also have lower upper part of the embryonal axis is epicotyle, and bottom part, lower part of the embryonal axis is hypocotyle. We are also able to observe radical, around which we have root cap cells. Now, with this, we have understood development of dicot embryo with graphics, with some diagrammatic representation, and we have seen that there are different stages in the development of dicot embryo, which are: first, there is development of proembryo from zygote by division of, by mitotic division, or by mitosis, or by equational division, and formation of two cells. One is bigger basal cell and the other one is smaller terminal cell, which will be later your terminal cell will be forming embryo and your basal bigger cell will be forming suspensor. And in a way, there is development, mitosis undergoing, and there is cell differentiation and development of embryo occurring, whereby there is entry of, there is a characteristic globular shaped embryo appeared at a stage, and that is called as globular shaped stage of embryo, which later develops into heart-shaped embryo, and which will be developing in, which will be entering into torpedo-shaped embryo. Later, torpedo-shaped embryo will be undergoing walking stick stage of embryo, and finally there is formation of mature embryo. And embryo is the source of new plant. Embryos are found within the seed of plant. This is a diagrammatic representation given in your NCERT textbook, wherein there is development of proembryo from zygote, which will be entering into globular embryo, which later develops into heart-shaped embryo. You can see the suspensor cells. Main function of the suspensor cell is it helps in pushing the embryo to within the endosperm, so that embryo receives nutrients and undergo growth and development. And this is your walking stick stage of embryo, or you can say this is mature embryo wherein you are able to find plumule, radicle, and two cotyledons. So presence of two cotyledons is the characteristic feature of your dicot embryo. Now these are the texts, these are the summary about dicot embryo development. So you can pause the video and read the text. So this is all about development of embryo in dicot plants. I'll be coming up with development of embryo in monocot plants. Sorry, I'll be coming up with another video regarding your development of embryo in monocot plants. So this is all for this video. Thank you. Take care. Have a good day.